Dear students, today I am going to discuss about the topic applications of monoclonal antibodies, that is MABs, in biomedical research, clinical diagnosis and treatment. This particular topic belongs to module 4 of your course MMB203. Monoclonal antibodies are an old immunological tool with the applications in the fields of immunology, basically, but in the field of biotechnology, in the field of biochemistry, applied biology, diagnostic microbiology. Monoclonal antibodies nowadays is widely used. So you can find that recently monoclonal antibodies have been widely applied in the field of clinical medicine. And 1/3 of all the new therapeutic treatments for breast cancer, leukemia, arthritis, then transplant rejection, asthma, psoriasis, with many more late-stage clinical trials being conducted with monoclonal antibodies. That is because humans have gained the ability uh, due to prolonged research to make antibodies able to bind to virtually any antigenic determinants, that is the epitope, to discriminate between two similar epitopes. So this remarkable specificity of antibodies makes them promising factors or promising element for human therapy. Now we have to discuss what are the problems. So the problems to be solved before the application of MABs before the use of MABs in human therapy, we have to counter some problems. That is polyclonal response. What is polyclonal response? The antibody can recognize and bind to many different epitopes of a single antigen. So that problem should be solved. And second one is a limitation of growth potential. These two problems should be solved. So we need something. So what are the needs? The needs are to make antibodies of a single specificity. Means a particular antibody will identify a particular epitope of a particular antigen. To make antibodies all built alike, manufactured by a single clone of cells. And to make antibodies that can be grown indefinitely. These are the targets. Sorry, these are the needs. And on the basis of our need, we have set some target. So target is to make an antibody that will bind only to one type or group of cells in a patient. Coupling a cytotoxic agent means we can couple along with the antibody a cytotoxic agent that may be a fluorophore, that may be a strong radioactive isotope to the antibody. And this radioisotope or this fluorescent probe will tells us or will function as a signature molecule, as a guiding molecule. Then giving the complex to the patient so it can seek out and destroy the only particular type of cells, not the normal cell. So the specificity should be such that the antibody will only bind to one type of or one group of cells, that may be the disease cells, that may be the infected cells or the cancer cell, not the normal cell. And the antibody should be coupled with a signature molecule that may be a strong radioactive isotope, that may be a fluorophore. It will guide us show that the antibody has bound with a particular type of cells, not with the normal cells, not with all sorts of cells. So this particular complex, giving the complex to the patient, so it can seek out and destroy only a particular type of cell. Now, discovery of somatic cell hybridization. This is very important discovery in the field of immunology, in the field of clinical biology. Because in 1970s, the B-cell cancer myeloma was discovered. And then it was understood that these cancerous B cells all produce a single type of antibody. This was used to study the structure of antibodies, but it was not possible to produce identical antibodies specific to a given antigen. So this problem was resolved 5 years after by the three scientists, Kohler, Milstein, and Zerini, and they have devised a technique. They have identified a technique, new technique, developed a new technique that is called somatic cell hybridization. And this particular technique results in a hybridoma cell producing antibodies targeting to a single epitope in large numbers. So here you can see what is the somatic cell hybridization. Here Sendai virus is used along with, here you can see Sendai virus is used along with, sorry. Here, sorry. Here you can see this. This is Sendai virus. So Sendai virus is used along with human fibroblast, this blue chromosomes, I mean from the origin, human origin, and mouse tumor cell, the red one. So under the influence of Sendai virus, human fibroblast and mouse tumor cell grown together in a culture, and there will be formation of hybrid due to the fusion, because the cell nucleus will fuse and they will produce a heterokaryote. So here you can see, due to the formation of, due to fusion, they are forming a heterokaryote. Eventually a number of hybrid cell lines will be formed. Here you can see, so cell line 1, 2, and 3 are A, B, and C, where colony type A, they are the hybrid colony. Containing means they are the hybrid colony, and here you can see these hybrid colonies here these hybrid colonies. Colony A contains only one, and colony B and C contains few human chromosome. So blue color chromosome you can see here in the picture. In colony A, there's only one blue color chromosome is there. In B, 2. In C, 3. So the number is reduced here. So by the formation or by the fusion, formation of fusion between the two type of cells, eukaryotic cell, in presence of, in the influence of Sendai virus, here 3 different type of colonies are formed. One is a hybrid, all are hybrid, but first one contains only one human chromosome, which are readily identifiable. So these cell lines can be screened for the presence of various human biochemical markers, which are then assignable to a retained chromosome. And these are the 3 scientists, Jerini, Kohler, and Milstein, got Nobel Prize in 1984. So one is, it should be one. So in 1984 in medicine and physiology. Monoclonal antibodies are monospecific antibodies that are the same because they are made by identical immune cells that are all clones of a unique parent cell. They have a monovalent affinity in that they bind to the same epitope. Given almost any substance, it is possible to produce monoclonal antibodies that specifically bind to the substance that can then serve to detect the substance. Monoclonal antibodies have become essential tool in unraveling the role of many genes and their proteins products in tumor pathogenesis and in the diagnosis and therapy of many diseases, including human cancer. So these are the importance of monoclonal antibodies in clinical or in human therapy. However, mostly the mouse monoclonal antibodies produced using a traditional albutoma technology are highly immunogenic in cancer patients, leading to HAMA response. What is HAMA? HAMA is known as a human anti-mouse antibody response. So resulting in their rapid clearance due to HAMA response, it will be rapidly cleared from the patient's serum. So technological advances in genetic engineering has developed a new type of antibody to reduce the immunogenicity so that it cannot be cleared very easily from the human body. So to reduce the immunogenicity and to increase the serum half-life of the rodent antibody in human by producing this type of antibody, which is known as chimeric antibody. Here you can see picture B. This is a chimeric antibody. Means the blue part is a human origin and the red part is a mouse origin. So this is chimeric, and the C, that is the humanized version of such antibodies. So one is a chimeric one, another one the humanized one. Next, fully humanized form, that is the D form, fully humanized monoclonal antibodies against human antigens have been developed by using transgenic mice containing human immunoglobulin gene or via first display technology. So biomedical and technological advances have facilitated the development of bispecific monoclonal antibodies, that is a type E. Here you can see this is bispecific, means here two different origin is there, bispecific antibodies with two distinct antigen binding sites. Here you can see two distinct antigen binding site is here, two distinct antigen binding site is there. So antibody drug conjugates as well as small antibody fragments. Here you can see F and G. These are monovalent single chain variable fragment and bivalent single chain variable fragments are having molecular weight of 30 and 60 kD. So they are used for the therapeutic applications and for cancer imaging. This particular type of antibody drug conjugates are used. This is how the monoclonal antibody can be used for different purposes, for therapeutic applications, for diagnostic purposes, for clinical applications, and for future applications. And all the antibodies has been approved by FDA, Food and Drug Administration, for human use. So for therapeutic applications, for say transplant resection, then Neuronomab along with CD3 can be used for cardiovascular disease. For cancer, rituximab can be used. For inflammatory disease, infliximab can be used. For cardiovascular disease, abciximab can be used. So these are the drugs that can be used in different for different therapeutic purposes. For diagnostic purposes, say biosensor development, microarray technique, this monoclonal antibody can be used for purification purposes, purification of drugs, purification of molecules, protein molecules especially can be used. And for future applications, we fight against bioterrorism. This particular type of monoclonal antibody can be used, and these are listed here in case of anti-inflammatory action, in case of cancer, in case of different type of cancer. What sorts of monoclonal antibody can be used? Here you can see rituximab can be used for non-Hodgkin lymphoma. Then trastuzumab can be used for breast cancer, etc., etc. So whatever the monoclonal antibody I have authored as and finished as MAB, so they have some nomenclature. We have to follow the nomenclature. And what is actually the nomenclature? So every monoclonal antibody has the following components in its name. First one is a variable one, that is written in red. Then is a target substance, right? For what purpose? Then the source substance, from where it has developed. Then stem, then additional word. This is for a special case. So in case of elafibranor, pigol, gol is additional word. So here you can see the red one is ala is a variable part. Then C is a target substance. Then Zu is a source substance. MAP is a stem, means monoclonal antibody. And Pijol is a additional word. So this is the target substance. It can be named as the old name and a new name. Two formats it's there. So NEB, Ba, Si, Feng, Kai, Lai, Ni, Os, Tox, etc. These are the new target substance. And this can be used in for the following diseases. Means NEB means angiogenesis inhibitor. Ba means bacterium against interleukin and inflammatory relations, immune systems, etc., etc. For tumor, these two for acting against tumor. Then what is the source substance? Means if it is coming from the rat, then the letters will be A. If it is coming from hamster, letters will be E. Primates I, mouse O, human U. And if it is chimeric, then it is Xi. Then it is, if it is humanized, then it is Zoo. It may be a rat mouse hybrid. It is Axo. So this is the source substance. And the example is here. You can see rituximab. So re is the variable part. 2 means acting against tumor. Xi means a chimeric one. MAB means a monoclonal antibiotic. Here additional part is absent. So rituximab is a chimeric monoclonal antibody targeting a tumor. Now, what are the advantages of application of monoclonal antibody? The advantages are homogeneity. It's a single antibody molecule that binds to antigens with the same affinity and promote the same effector function. Next is specificity. Means single hybridoma reacts with the same epitope of an antigen. Then immunizing an antigen means need to not be characterized, and it is ultimately not needed in large quantities to produce large quantities of antibodies. Highly sensitive. Then selection, select for specific epitope specificities and generate antibodies against a wide range of antigenic determinants. Next is antibody production, unlimited quantities of a single well-defined monospecific reagent can be produced. Now, what are the disadvantages? Disadvantages are average affinity. Second one is may lose reactivity with antigen. Then the third one is display unexpected cross reactions, means some toxicities there with unrelated antigen. Then immune rejection that we can find in transplant rejection, immune rejections. Time and effort commitment, very large, very expensive to produce. So these are the disadvantages of monoclonal antibody. Monoclonal antibody can be used for diagnostic purposes. So they're mostly used for the following tests: Western blot analysis, ELISA test, antigen capture assay, then immune dot blot test to detect a specific protein on a membrane. Then in case of immunohistochemistry for histologic tissue section to detect antigen present in a tissue section. Then immune fluorescence test to detect presence of antigen in a cell. We can detect immune fluorescence test via immune fluorescence test. So for this particular test, we can use monoclonal antibody either solely or conjugated with some tracer molecule, that is maybe some radioactive isotope or maybe some fluorophore. But if we are using radioactive isotope in the technique of RIA, radioimmunoassay, in case of simple ELISA, enzyme linked immunosorbent assay, to simply use some chemical, or immunofluorescence assay, we can use some fluorescent molecule. So these are the tests, diagnostic tests used for the diagnosis purpose, where monoclonal antibody can be used. So number of monoclonal antibody has been developed for the detection of diseases, for the diagnosis of diseases. These are the listed diseases. So it may be a parasitic disease, Trichomonas vaginalis, Leishmania, Donovan, Trypanosoma, congolens. We can use monoclonal antibody for their detection. For human viruses like influenza virus, rotavirus, rabies virus, etc. For animal viruses, say bovine herpes virus, say cervine herpes virus type 1. For this particular type of viruses, for this particular type of parasitic infections, identification or diagnosis purpose, we can use monoclonal antibody. In case of pregnancy test, we can also use monoclonal antibody. It's a kit-based test. Monoclonal antibody can be used to detect whether pregnancy is there or not, with antibody to beta human chorionic gonadotropin, beta HCG. So here we have to collect urine sample from the female, and then first early morning uh, urine sample we have to collect, and we have to put one drop at a particular space. Here you can see, here we have to put only a single drop is sufficient. It's a very sensitive test, a single drop. Here we have to use, and this is a control one, this is a test one, and this showing no mark here, so it's a negative, and here you can see some mark, so it is test positive. Here you can see this immunoglobulin or the monoclonal antibody to HCG. Color beads are also there, so they will cross and they will react here. So second antibody, here you can see they will react here and will develop the color here, will develop the mark here. Here you can see the color mark or the band we can have. So this band specifically shows that whether it's pregnant or not. In case of malarial parasite card test, we can also use it, a similar type of test, card-based test. So capture the parasite antigen from peripheral blood using monoclonal antibodies prepared against a malaria antigen target, and conjugated to either a liposome containing selenium dye or gold particle in a mobile phase. And a second or third capture monoclonal antibody applied to strip of nitrocellulose. Here the strip contains nitrocellulose, acts as a immobile phase. So it's a immobile phase or the stationary phase is nitrocellulose. The mobile phase is used here, containing a liposome containing selenium dye or gold particle. So the migration of the antigen antibody complex in the mobile phase along the strip enables the labeled antigen to be captured by the monoclonal antibody of the immobile phase, thus producing a visible color mark there. and will show whether the patient sample is malaria parasite positive or not. In case of Micral test, this is an immunoprecipitation test. Also, we can use monoclonal antibody. This particular test is based on color shift of monoclonal antibody to human albumin labeled with gold. Here you can see it is labeled with gold, and you can have some mark there, like the first one, the second one. In case of immunodiagnostics, as immunodiagnostic, you can use for separation or for purification of some substances. You can use monoclonal antibodies. Can also be used to purify substance with techniques called immune precipitation and affinity chromatography. So this is for the purpose of immune diagnostic. Monoclonal antibody can be used in Western blot or immunoblot. Monoclonal antibodies are used that helps to identify a specific molecule or protein substance that is in a mixture, whether it's present or not. So it can identify in very less quantity. Thank you.